my name is Ianiri, and I am an ocean sciences PhD student at the University of California, Santa Cruz. My undergraduate degree is in chemistry with minors in environmental studies and Spanish from Northeastern University. I also completed internships at Woods Hole Oceanographic Institution and the Chesapeake Biological Laboratory, which is where I became inspired to pursue a career in chemical oceanography. For my graduate thesis, I studied marine dissolved organic carbon. Dissolved organic carbon acts as a capacitor between atmospheric carbon dioxide and long-term carbon storage in sedimentary rock over millions of years. The marine dissolved organic carbon reservoir contains about as much carbon as all atmospheric carbon dioxide, meaning any major changes to the size of this reservoir would have drastic implications for our climate. But oceanographers don't really understand how marine dissolved organic carbon is going to respond to climate change. Dissolved organic carbon is often thought of as two separate fractions. A small portion is recycled in the surface ocean, where it serves as the base of marine food webs and is pretty quickly recycled. However, some organic carbon escapes this short-term cycle and accumulates in the ocean as inert organic molecules. These molecules are on average thousands of years old, meaning they're sequestered from the atmosphere on human timescales, and they make up the majority of the marine dissolved organic carbon reservoir. Still, despite decades of research, oceanographers don't know why these molecules persist for so long. Recent work suggests bacteria may be responsible for forming stable organic carbon molecules. A breakthrough which would imply bacteria are actually responsible for how the marine dissolved organic carbon will respond to climate change. For my graduate thesis, I explore this connection between bacteria and long-term carbon storage. To do this, I analyzed a unique set of dissolved organic carbon samples. We collected thousands of liters of ocean water, filtered out the organic carbon, and separated it into old and young dissolved organic carbon. This novel sampling process allows me to specifically investigate what is unique about the old organic carbon which accumulates in the ocean. I measured molecules produced only by bacteria, or bacterial fingerprints, and compared the concentrations in old and young dissolved organic carbon. Additionally, through collaborations at Lawrence Livermore National Laboratory, I'm using state-of-the-art instrumentation to develop methods for investigating the unknown chemical structures of old and young dissolved organic carbon. Together, these approaches will help us understand what processes form stable organic carbon molecules, with an ultimate goal of unraveling how the oceans will respond to changes to the global carbon cycle. Thank you for listening.